Hi everyone, welcome to my channel Pure Biology and with another video obviously, and this is the video of about DNA binding motifs, and there are four kinds of motifs. So two are already explained, so the remaining two have to be explained now. So third one is the leucine zipper motif, and the fourth one is the zinc finger motif. Okay, leucine zipper motif and zinc finger motif. So leucine zipper motif, that mediates both DNA binding and dimerization process. That means its role is to continue the DNA binding process and obviously the dimerization process of the protein. And the two distinct right-handed alpha helices that participate in the formation of the homo and the heterodimer structures. So here also two helices are present, alpha helices are present, that help in the formation of the heterodimer and the homodimer structure. And N-terminal DNA binding domain is rich in positively charged amino acid residues, and the C-terminal dimerization domain. So here main important thing is that there are two terminal region. One is, this is the rough structure, this is the N-terminal region and this is the C-terminal region, and the N-terminal region, this is called the DNA binding domain, and this C-terminal region, this is called the dimerization domain. Okay, so there are two parts, one for the DNA binding domain and another is the dimerization domain. Now leucine is very important here, because leucine is one amino acid, and this is why it is important here, because the leucine zipper domain is necessary. No, this is not leucine at every 7th position. That means here the 30 to 40 amino acids are there. I just have drawn a very short structure because the space is very short here. Just I try to explain you that here 30 to 40 amino acids are there, and within 30 to 40 amino acid, the leucine must be present at the 7th position. Okay, leucine must be present at the 7th position. So that's why it is called the leucine zipper domain. Clear? This is necessary for the protein dimerization. Already I told you. And the two alpha helices in parallel wind around each other to form a coil structure. So here this is very simple word, that the two alpha helices, they form a coil. Okay, and in that way, or in the coil form, they remain. The leucine residues end up with their R groups protruding from the alpha helical domain in which the leucine residues reside. So one important thing is that this protruding area, this is a protruding area that continuing one R groups. Okay, one each contains with one R group. Why this one R group is important? Because when it attached with another one, these R groups that enters with the another alpha helices R groups, or make a bond with the another alpha helices R groups, or R groups of another alpha helice. Okay, so that's why these R groups are very important, and the protruding R groups are thought to interdigitate with the leucine R groups of another leucine zipper domain, and thus stabilizing in that way. That means due to this attachment, the stabilizing homo or hetero dimerization is possible. Clear? So here these R groups and the leucine groups both are important here, because these R groups of this leucine domain is why important, because this attached with the another R group of another domain and forming a stabilized structure. So this is the leucine zipper motif. The last one is the zinc finger motif. Here the leucine was important, and here the zinc is very important. There are two kinds of zinc finger motifs. The First one, where the structure is seen generally in that form, C2H2 form or CYS2HIS2, in that form. This is the first structure, and the second structure, it is seen in the form of this CYS2, or you can say in that way C2. Clear? Now, zinc is very important. Why? Because it is zinc coordinated motif. Okay, this is the zinc coordinated motif, and this occurs in the clusters, and each is 30 amino acids. So here 30 amino acids residues are seen, and characterized by CYS and HIS that always occur at the same position. So in the first case, where the CYS and HIS present, so where the structure is seen generally, the position of the CYS and HIS that always be same, and it forms a compact globular domain in which a zinc ion is coordinated tetrahedrally by CYS and HIS. Okay, why it is called the tetrahedrally? Because 2 HIS and CYS2, both CYS and HIS that are 4 in numbers. Okay, that's why it is called the tetra. 4 are in numbers, and zinc is just attached with this HIS and CYS in between. That means 2 HIS and 2 CYS, they attached with one zinc ion. Clear? That's why it is called the tetrahedral structure. The domain is composed of 12 residues alpha residues means here amino acids, 12 amino acid residues. Okay, amino acid, and alpha helix packed against an irregular beta sheet with zinc in between. So here the alpha helix and this is composed with the beta sheet, and these alpha helix and beta sheet, these are the structures, and obviously these are the topics of biochemistry. So I will try to explain this beta sheet and alpha helix in another video. Okay, just here you just remind it that both alpha helix and the beta sheet both are composed here, and the alpha helix lies in the major groove. In the same way, alpha helix that always present within the major groove of the DNA and contacts with 3 base pairs. So the difference is what? Difference is what in between the C2H2 and C2C2. Here the HI is present, but here in this case HI is absent. Only CY is present, and here also the tetrahedral structure is seen, because here the 2 HIS and 2 CYS that form a tetrahedral structure, but in the second condition the 4 CYS that make the tetrahedral structure, and here the amino acids number is 70 to 80 residues, and this contain the 2 zinc ions and each coordinated tetrahedrally with 4 CYS residues, and the number of zinc ion here it is 1, but in case of second one I haven't drawn here, so there 2 zinc ions are present and 4 CYS residues are present. So these are the structure or you can say the factors related with the eukaryotic transcriptions, because these are also involved in the attachment process or anchoring process. So these factors are very important related to the eukaryotic transcription, and you just check out the eukaryotic transcription process from my channel, because it is already present there.